Molecular biological tools, or MBTs for short, allow us to detect and quantify specific microorganisms and genes of interest in environmental samples, including groundwater. MBTs have been refined in recent years and are being used for site assessment and the identification of sites where bioremediation can be implemented as a productive remedy, and are also used to monitor the progress of bioremediation.
Remedial program managers and regulators want to know, how long is it going to take to clean up this site? Design engineers typically answer this question by extracting a rate constant for degradation from the monitoring data and then using the rate constant to estimate the time when the cleanup goal will be reached. Many sites don't have enough data from wells that are in the right place to be able to extract a useful rate constant. If we could use MBTs to predict a rate constant for degradation, we should be able to apply monitored natural attenuation at many more sites.
Commonly applied MBTs measure nucleic acids, which link indirectly to biodegradation activity, but these measurements cannot inform about degradation rates. A more direct measure of activity would be to quantify the catalysts, in our specific case, the reductive dehalogenase enzyme systems. We take advantage of the advanced analytical capabilities of state-of-the-art mass spectrometry instrumentation, specifically proteomics, and quantify specific reductive dehalogenase enzyme systems in groundwater.
During the last several years, our team has developed protocols for the extraction, digestion, and analysis of proteins from complex environmental samples, including sediment, soil, and water. Following extraction, proteins are digested, purified, and analyzed via advanced mass spectrometry. Furthermore, our advanced software can be used to identify signatures and/or proteins of interest. We also provide targeted identification and quantification of specific proteins of interest in environmental samples, such as groundwater, sediment, and soil. All of these capabilities are now available to our commercial and governmental clients through collaboration with Microbial Insights.
By knowing what these enzymes do, and by determining their absolute abundances in groundwater, we are able to determine if enzyme abundance can be used to predict biodegradation rates. In other words, our approach is to correlate the abundance of specific protein biomarkers with degradation rates, which will provide crucial information about the cleanup time required to meet regulatory standards. Such a rate predictive tool would transform how we assess monitored natural attenuation and bioremediation, and would make remediation outcomes much more predictable, which will streamline site cleanup.